Hey, Matthew Tyler and David Roy, my two stalwarts. You guys are always here first. Is one of you in a room with a bunch of people?
Believe that would be Matthew.
Yeah.
Okay.
But this on Mondays we're in the room where the computer doesn't necessarily set up. So
Okay. Well, I think I was talking to Dr. Lounsbury. She said that you guys might want me to come in and give live lectures. I'd be more than willing to do that. We just need to find a time and a place. So I'll get back to you that through Brightspace this weekend. I'll let you know what's going on with that, because maybe as soon as Monday we can start doing that.
cool.
This might be a dumb question, but would you still be recording?
Yes, absolutely. She said there's a room where it can record easily. I don't know. I got to move. I move around a lot, but we'll see.
That'd be a very long drive for me to come up there.
You don't have to. You don't have to. Hey there, Patrick. Good to see you again. Kaylee's here. Life is good. Hannah Moynihan. Moynihan. You know, when I was in college, the Speaker of the House was Patrick Moynihan, Daniel Patrick Moynihan. I don't know if you know that guy, but when I graduated college, he was the speaker, the invited speaker at my graduation. Small claim to fame. So everyone needs to know a Moynihan at some point in their life. All right, so I guess
Dr. I had a quick question before we start.
Okay.
I was
go ahead.
under the impression that we finished chapter 9's notes, and I didn't notice anything posted to Brightspace, so I didn't know what we were doing today.
Right. What I posted last week was not complete. I'm in the process of writing the notes. So I reposted yesterday with what I've completed since then. And we're going to finish chapter 9. We're going to go through the rest of chapter 9. I don't know if I'm going to finish it today, but all the notes are there for chapter 9 now and the PowerPoints. So
Thank you.
This is a work in progress. Sure. Zoe showed up. Does it annoy you when people call you Zoe? That would really annoy me.
No, only when people pronounce my name wrong.
What's the right way?
Zoe's the right way, but when people pronounce it Zoe, thinking that's the whole thing.
Yeah, that's annoying. That's annoying. Okay. All right. Well, let's go ahead and get fired up here. I want to do a quick review of what we did a week ago. If I can get to it here. Oh my God. Okay, wait a minute. Come on. Yep, not letting me do that. Let's just do entire screen. All right, so I'm sharing my entire screen now. Okay, can we see the receptor tyrosine kinases?
Yep.
Okay, good. So I believe this is where we started a week ago. It's been a week and a lot has happened since then. But basically, we started out talking about the PI3 kinase mechanism. And the idea here is that PIP2 in the membrane, we've talked about quite a bit. It can be phosphorylated by PI kinase, which makes PIP3. PIP3 then is a binding site, or your binding site for any protein that's got a plextrin homology domain. And a lot of kinases do. In particular, we talked about PDK1, PDK2, and AKT. So PDK2 is called mTOR, mammalian target of rapamycin. Oh, I throw this in here just for a little levity, perhaps. It was the drug rapamycin was found on the island Rapa Nui. Anyway, so that's what this mechanism is kind of. What do I call it? The recruitment strategy that we see in a lot of these tyrosine kinase type mechanisms, where you make a binding site and that brings a bunch of proteins in based on their binding sites, and now they're going to phosphorylate each other. So you're bringing proteins near their substrates, and that's what kind of activates the pathway. The other part of this pathway is the destruction complex. Now, if you look this complex up, on Wikipedia or do Google searches or whatever, You can find some variation in what makes up the destruction complex. So don't get too uptight about that, because like here I've got WTX, and in other places you'll see different proteins being part of the complex. What's most important to know about this complex, especially in this mechanism, is GSK3 beta, APC, and axin. Axin especially will come in the Wnt process. So anyway, GSK3 beta is a kinase, and it's constantly tonically phosphorylating beta-catenin, and that causes it to be ubiquitinated and degraded. And again, we're going to talk about ubiquitination eventually. We just can't do it all at once, so we will get to that. But anyway, tonically destroying beta-catenin. So what happens in this mechanism is PI3 kinase gets recruited to the membrane by its SH2 domain and starts phosphorylating PIP2. That brings these other kinases up, PDK1 and mTOR phosphorylate AKT. AKT then inhibits GSK3 beta, allowing beta-catenin to build up in the cell. Beta-catenin is going to go into the nucleus, it's a transcription factor, and is going to activate transcription of genes that are going to cause the cell to go into the cell cycle. Cats near the end of their life start doing this. This guy's 20 years old and he's a bit of a loud guy. Anyway, I don't know if you can hear that cat, but bottom line being, when you activate AKT, you're going to activate cell division. And the other thing I talked about was how when you activate cell division, there's a bunch of other things you have to do. You can't just turn on cell division. One of them is inhibiting apoptosis. And in this slide from your book, you can see all the different pathways involved with AKT that are important for cell division. Here we are activating cell division itself by upregulating c-Myc and cyclin D. We'll talk about these guys in the next chapter in cell division. We're also going to inhibit CDK inhibitors, which we'll talk about in the next chapter. So this is all involved in increasing cell cycle division. We're also activating metabolism. GSK3 beta we talked about in terms of the insulin mechanism. So it's also involved in metabolism. And we're blocking apoptosis by blocking FOXO, which upregulates Fas ligand and BIM. And we're directly inhibiting BAD. These are all involved in the apoptosis pathway. Again, we'll talk about later. And MDM2 regulates P53, so this is also involved in the apoptosis pathway. And the third kind of major stream we're turning on here is increasing protein synthesis. So through this whole pathway here, we're going to increase the rate at which ribosomes translate mRNAs. And you can see we've got in the middle of this mTOR here and an mTOR over here. These are isoforms of each other, and they each have different roles. I think this mTOR is actually rapamycin insensitive, and this is the one that rapamycin works on. And rapamycin, like I said last time, is an immune suppressor that's used in a lot of transplants and things like that. Okay, so we're activating metabolism, cell proliferation, cell survival, and protein synthesis. And this is all going to help that cell to start dividing really quickly. And then I went into the insulin mechanism. I actually have an animation of this, but I need to translate it from Flash into HTML5. I'm still working on that. But bottom line is that it starts out the same way, except GSK3 beta, where is it? Right here. GSK3 beta is inhibiting glycogen synthase. When the insulin binds the receptor, that brings up insulin receptor substrate 1, which gets phosphorylated, has SH2 binding domains, that brings PI3 kinase up, activates AKT through PDK1 and 2. AKT then inhibits GSK3 beta, so now you can have glycogen synthesis. And AKT will also phosphorylate a protein that will cause fusion of this subcellular vesicle containing a glycogen, a glucose transporter. So now you've got glucose transport happening faster and glycogen synthesis. So insulin causes you to take glucose into your cells and store it as glycogen. And we keep coming back to this. We'll see it again today. Breakdown of PIP2 to produce IP3 and diacylglycerol. So there's an isoform of protein kinase C, which is PKC gamma, which has got an SH2 binding domain. And these receptor tyrosine kinases will bring that up as well, producing IP3, releasing calcium, activating or releasing diacylglycerol, activating protein kinase C, which also needs calcium. So the mechanism kind of is synergizing itself there, I guess. And we'll see that again in a heartbeat here, in a couple heartbeats, several heartbeats. Okay, so summing it all up, receptor tyrosine kinases, you've got the whole GRAB2, SOS, RAS, RAF, MEK, MAP kinase pathway. And then you've got the PI3 kinase pathway that goes through AKT and GSK3 beta. And then you've got the IP3 calcium mechanism over here. And remember, those are just a few of the many, many things that get activated by these receptor tyrosine kinases. Here is slide number 4. You can see when a receptor tyrosine kinase gets activated, there are a lot of SH2 binding domains that get made, and a lot of things are going to get recruited to the membrane and activated. Okay. So the next one we went over is the JAK-STAT pathway. Really simple pathway, starts out almost the same as the RTK pathway. The only difference being that the kinases are separate proteins from the receptors, but they bind to the receptors there and then they get activated and they transphosphorylate. And this is the neat part for me anyway of this mechanism, the way the stats come into play. So each stat has got an SH2 domain and an SH2 binding domain. So the SH2 domain brings it up to the receptor, where it then gets phosphorylated by the kinases, gets released, and now it's got both domains, so it binds to itself. You get a heterodimer of STAT1 and STAT2, and this becomes a transcription factor. Very short pathway, very, very effective, very efficient. I think cytokines go through this a lot, so this is really important in the immune system. And then we've got our serine threonine kinases, TGF-beta-like signaling, where you've got the big change here from the other is that the kinases are serine threonine kinases, not tyrosine kinases. And you've got two different types of receptor. You've got the type 1 that binds the hormone and the type 2 that then binds to the type 1. And they all clump together just like we saw in receptor tyrosine kinases and cross-phosphorylate. Now, similar to the JAK-STAT, I guess, you've got a receptor SMAD that's going to come up and get phosphorylated. And this SMAD will then bind to the cellular SMAD, SMAD4. What's kind of a little, one thing you should know, let me just focus on this for a sec, is that there are quite a few, not quite a few, I would say at least a dozen hormones that work through this system. I could be wrong with that number, so please don't quote me. And some of them go through SMADs 1, 5, and 8. And others go through SMADs 2 and 3. So these are the receptor SMADs, the ones that get phosphorylated by the receptor. BMP4 activates SMADs 1, 5, and 8. TGF-beta activates 2 and 3. So, and why that's important is when you get into embryology and embryo development, you'll see that you've got both BMP and nodals, and they're activating different genes because they go through different receptor SMADs. So that's kind of an important distinction. So you get different genes being turned on based on which SMADs are being, which receptor SMADs are being activated. But the cellular SMAD is always SMAD4. Okay. And then Wnt signaling. I bring up colon cancer because the production of new epithelium in the large intestine is based on Wnt signaling, which causes cell division. And as those cells divide, they push up out of the crypt. And then they, at this point, Wnt signaling stops and they differentiate into surface epithelial cells. And then they die up there and they get replaced from behind, constant renewal. So this is why that one protein in the destruction complex is called APC, because if you don't if you can't turn off this Wnt pathway, you get proliferation of cells and eventually invasion and eventually metastasis. So Wnt signaling is really important in colon cancer. And then that's where this guy comes in, APC. So we saw APC previously with the destruction complex. The name is, and I got to say this name because it took me 5 years to learn how to pronounce it correctly, Adenomatous polyposis coli. Adenomatous. I don't know why that word is so hard. But anyway, that's just a structural protein that's important in holding the GSK, the destruction complex together. Now, as I said before, you look at different sources, different tissues, different species, you'll see different proteins being involved in the destruction complex. And we pretty much always have APC and axin. WTX is not in it this time for some reason. I don't know. DVL is disheveled up here. And disheveled is going to be Wnt specific. And axin is also important here. So, and something else that's been thrown in here, now you've got casein kinase as well as GSK3 beta. And another thing they've put into the complex here, which we were just assuming before, is your ubiquitinating enzyme, beta TRCP. So GSK3 beta and casein kinase phosphorylate beta catenin, and then the ubiquitin ligase here puts ubiquitin on it, and then it goes to the proteasome and gets degraded. So it's all part of one big destruction complex, constantly breaking down beta catenin. Now here's where the Wnt signaling is a little different from the PI3 kinase signaling, in that when Wnt is around, it binds to its receptor, which is frizzled, and then it also brings in its co-receptor, LRP. And then that binds disheveled, and disheveled then technically brings axin off this complex, which breaks up the complex, and now beta catenin is free, and we're activating cell division again. So bottom line here, frizzled and LRP are the co-receptors. When Wnt binds it, it binds disheveled, that brings axin up, and that ruins the destruction complex. Beta catenin. Okay, and non-canonical Wnt signaling, we're doing phospholipase C and IP3 calcium again. And so this, the receptor complex can activate this G protein. And this is what we're going to talk about today, so I'm not going to talk about it now. And then the non-canonical planar pathway. And what I like about this is it kind of explains chemotaxis, in that when you've got Wnt around, cells are attracted to it, and they do that by bringing up disheveled. Disheveled then activates the G protein that activates RAC, and then it also activates the disheveled activated, what is it? Disheveled activated modulator of, disheveled associated activator of morphogenesis. So this complex here is activating Rho and RAC. And we saw RAC and Rho back in chapter 3 when we were talking about cell crawling. So here we can see a chemotactic hormone, Wnt, causing chemotaxis in a cell. So it's going to cause the cell to remodel its actin cytoskeleton and move. Hedgehog signaling. Did we talk about this last time? Anyone remember hedgehog?
I don't think so.
No? Okay. So I'll go a little slower here. This is another hormone that's really, really important in embryo development. It's used a lot for patterning in the early embryo. So the way it works is you've got a receptor for hedgehog in the membrane, which is called patched. And patched is inhibiting smoothened tonically, okay? So in the off state, we're inhibiting smoothened. Now here in the center, we've got, again, kind of a destruction pathway, in that we've got it's not the same pathway, but similar, in that you've got a protein here called GLI. They call it CI GLI here because it depends, again, on what species you're looking at. In Drosophila, it's called CI. In mammals, it's called GLI. So anyway, this protein is interesting in that it's got two domains. One domain binds to DNA, and the other domain is called the activation domain. And what it does is bring other proteins to the DNA once it's bound there. And you probably know, at least I hope you know, how genes are activated, which is proteins are brought in and they bind to the enhancer, and then that causes the bending of the DNA strand, which will go back to the promoter, and that brings the RNA polymerase in and everything. So getting proteins together on that DNA is what activates the gene. So anyway, you've got this protein which is half DNA binding and half other protein binding, and it needs to be whole to be a transcription factor. Now, in addition, we've got this guy here called fusion, and this called, is it suppressor of fusion, I believe, suppressor fusion, SUFU, suppression of fusion, and COS2. So COS2 is got, see, it's an isoform of kinesin. Remember kinesin, again, from chapter 3, it walks on microtubules. So we've got a microtubule binding protein here that's binding to GLI and also fusion and sonnefusion. So these are all binding together, and COS2 is keeping it on microtubules. Now, what your book doesn't tell you, but you'll see in just about everywhere else, this is in what's called a primary cilium. Primary cilia is like a cilium, but it's used for signal transduction and not for movement. So it's not a movement cilium, but it's structurally very similar, I guess. So anyway, this whole complex keeps GLI up into that primary cilium, and patched inhibits smoothen. Smoothen will bring it out of that cilium, but patch is tonically inhibiting it. So in the unactivated state, you've got this complex, and because it's complexed, it causes phosphorylation of GLI and then proteolysis of GLI. So only the DNA binding chunk of GLI comes out, it binds to the DNA, and then it acts as a repressor because it doesn't have the activation domain, it just has the DNA binding domain. So it's, you know, like an antagonist. It's binding to the site, but it's not allowing activation. So that's a repressor. Okay. So you ask me, what happens when hedgehog comes along? Hedgehog will bind to patched, and it's going to stop patched inhibition of smoothened. So the smoothened then moves up into that primary cilium and releases this complex, which causes the whole GLI to be released unproteolized, and then that will go to the nucleus and activate transcription. Pretty weird transcription pathway, but I think it's pretty neat. So again, your hedgehog is going to bind to patched. Patched stops inhibiting smoothened. Smoothened goes up into that cilium, releases GLI, and then GLI activates target genes. Interesting. Again, I'm going to give you another interesting brush with history. So I was doing my postdoc at Harvard Med in one lab, and I had a friend in the Tabin lab at Harvard Med. This is like early 90s. And they discovered that there were several different kinds of hedgehog. One was called desert, one was called Indian, and this guy I knew found the third one. And because his son was playing a lot of video games, he saw Sonic the Hedgehog, so he called it Sonic Hedgehog. So that's why it's called that. So little brush with fame. I gotta say, I didn't do anything at Harvard Med School that would make me this famous. Okay, anyway, any questions about hedgehog? Kind of a neat pathway. Again, the key to this is that you've got this protein GLI that can be cleaved to become a transcriptional inhibitor or released free to make it a transcriptional activator. And here's an idea of what hedgehog does. You've got, it's being released from tissue, I believe right here, like right at the edge of the right at the edge of the limb bud, and it's forming a gradient out, and that hedgehog then is causing the differentiation of fingers. So based on your hedgehog signaling, you can get more or fewer fingers. So it helps to pattern that limb. Yay, hedgehog. Okay, notch signaling. Now this pathway is really simple. similar to the hedgehog, because it involves, because it involves proteolysis. But the example it's used in a lot of places in embryo development. The example in your book here is the production of valves in the heart. And the key thing here, which is important for Notch, is it causes this endothelial to mesenchymal transition. This is something that happens a lot in embryo development. It's really important. Basically, endothelial cells are connected to each other to make a sheet, right? That's the whole idea of endothelial cells. It's a sheet of cells. And they're connected to each other by cadherins. Chapter 3, we talked about integrins, which are transmembrane proteins that grab extracellular matrix. Here, these cadherins are transmembrane, but they grab cadherins, the same cadherin in the neighboring cell. So it's cell-to-cell junctions that are keeping these cells into a sheet. So E to M transition then is when the cell goes from that kind of static in a sheet structure to letting go of its neighbors and crawling. Really important in embryo development, cell crawling. So anyway, what's going on here is you've got this sheet of endothelial cells. Some cells get stimulated by Notch and they let go of the neighbors and they crawl down into this cardiac jelly, and then they form the valves in the heart. Also, I like to throw out embryology because I think it's really neat. Another thing Notch is important for is you've got this solid chunk of mesodermal tissue, and Notch signaling turns that into somites. And if you know anything about embryology, the formation of somites is important for body segmentation and the development of the body plan. So anyway, Notch is really important for that too. Okay, Notch signaling. I like to waste time talking about EMT because Notch signaling is so short, it's quick, it's easy. I'll be done in a second here. You've got it's juxtacrine signaling. You've got one cell with the ligand on it, another cell with the receptor. So the ligand is delta or jagged, and then the receptor is called Notch. And when jagged or delta hits Notch, you get proteolysis to cleave that receptor binding part out. And then you get two more cleavage reactions that release the N-terminal intracellular cytoplasmic domain of the receptor. So that's a transcription factor. That's all it takes. You just get these cells to touch together, proteolysis, release of a transcription factor. And that's going to go in and activate some genes like snail. And snail is going to decrease the production of cadherins, and they let go of each other. And then there's other genes being activated that are causing the cell to start dividing or start moving and crawling and stuff. So here, again, the E to M transition, endothelial to mesenchymal, endothelial to sheet, mesenchymal to crawling cells. E to M transition, cleavage of the transcription factor from that was part of the receptor, goes into the nucleus, and then turns on all those genes. Okay, so that's the end then of all the kind of morphogenic, mitogenic type signals, things that are going to activate genes and cause changes in transcription, changes in differentiation, and mitogenesis and all this stuff. Now we're talking about things that are a little more physiological. And it is a major transition because this kind of stuff was known already when I was in grad school, and everything we talked about so far was learned since I left grad school. So this is really much older science here and much simpler, I think, which is why it was done earlier. But anyway, that's just a little editorial content. So here we are, we're talking about G protein coupled receptors. G protein coupled receptors are shockingly ubiquitous. There are so many of them. The main ones I'm interested in, being a cardiovascular pharmacologist, are the ones involved in the sympathetic and parasympathetic system. But there are at least 831 genes. And at least one Nobel Prize, a woman, Linda Buck, who, again, I knew when I was at Harvard, won the Nobel Prize because she isolated a gajillion G protein coupled receptors and showed how they were they were the receptors involved in the taste and smell in the mouth and nose. So really important discovery there. So anyway, all autonomic signaling goes through this. Your vision and smell receptors go through this. You've got a lot of this in your CNS. There's just an awful lot of G protein coupled receptors. What they have in common is they all have this 7 transmembrane structure. They're often called 7 transmembrane receptors, at least they used to be. You've got an extracellular amino terminus and an intracellular carboxy terminus. And you've got 1, 2, 3, 4, 5 transmembrane regions, then this one long intracellular region, and then 2 more transmembrane regions and a really long intracellular tail. And what's neat about this is you can figure out where the activities are here. The ligand's going to bind here, kind of between this loop and this loop, really, somewhere down in the pocket here between those two. This then is the part that's going to activate the G protein. And then out here, we've got phosphorylation sites that are going to be involved in regulation. Regulation of receptors is going to be the last thing we talk about in this chapter. So anyway, you've got ligand binding domain and activation domain. Oops, what am I doing? Let's come back a little bit. Activation domain there, and then regulation domain down here. And when you look at it in a more three-dimensional view of it, the alpha helices come together like this. And right down in here is the binding pocket, right down inside here. And here's, I love this picture. This shows you the how this is norepinephrine binding to the beta receptor. You know, you've got serine doing hydrogen bonding here, another serine here, hydrogen bonding, aspartate binding to the protonated ammonia group there. So that's how this thing works. And you can imagine too, this is how you activate that receptor. When the hormone comes down into here, it's going to pull the top part of that together, which is going to open the bottom part of it. You get a conformational change, and that allows it to activate the G proteins. Oh, and speaking of G proteins, here we are. So we talked about G proteins quite a bit already in terms of your small monomeric G proteins, RAC, RO, CDC42, RAS. Those are all small monomeric. These are large heterotrimeric G proteins, so they're a little different. What's different about them is they always have this beta gamma subunit attached to it when it is in the inactive state. So in the inactive state, your G proteins are going to have GDP attached. When you get a GEF, a guanine nucleotide exchange factor activated, it causes it to release GDP and pick up GTP. And then your alpha subunit separates from the beta gamma. It goes off and binds to some effector and causes changes in the cell. Beta gamma, for the most part, is hanging out waiting there. Here's another difference between this and the small monomerics. This has got a lot of, this has got an active GTPase activity. The small monomerics have got very low GTPase activity, and they need GAP, GTPase activating protein, to stimulate it. Here, these actively break down GTP into GDP and release phosphate and turn themselves off. So you can imagine, we always call this G protein cycling, because if you've got a ligand on the receptor and you've got activated receptors out there, these G proteins are going to get turned on, turn themselves off, turned on, turn themselves off. So they're constantly cycling in this manner until you no longer have activated receptors, and then they stop in the inactive form. And here's just kind of a figure of that. Here's your generic receptor and your generic G protein. And ligand binds, activation of the G protein. G protein activates some effector protein, and then hydrolyzes GTP, and then it comes back together again. Okay, so that's your generic G protein figure. Now, what you need to know is there are many isoforms, of course, of G proteins. There are three main classes that we're interested in. There is GS, GI, and GQ. And each of them is going to be linked to a different effector or several different effectors and giving a different response. And they're going to be linked to different receptors and produce different receptor responses. So we'll start out the two main second messengers involved with these G proteins are cyclic AMP and the IP3 calcium that we keep talking about. And I like to think of them as sympathetic and parasympathetic, because when you get an autonomic physiology, pharmacology, that's what we're talking about. Sympathetic nervous system stimulates cyclic AMP, parasympathetic, it activates IP3 calcium. And we'll talk about shortly how those are two physiologically antagonistic to each other. Okay, so cyclic AMP then. Here's our old friend ATP, our good old friend ATP. So as you probably know from biochemistry, these squiggly lines mean high energy. Each of these is 7.5 kilocalories per mole. So it takes a lot of energy to put these phosphates on here because you've got all these negative charges you're putting next to each other. So there's a lot of energy stored in that. Now, the enzyme that's going to make cyclic AMP is adenyl cyclase. Adenyl cyclase then will take this phosphate and react it with this 3' hydroxide over here, or 3' carbon, and you get this circle, cyclic AMP. Now, one thing I want to impress upon you, one thing that's really, really important about this mechanism. Never call it camp, okay? You call it camp, you're going to break your pharmacologist's heart. It's called cyclic AMP. Okay, camp is where you go, you know, when you're a kid and you shoot bows and arrows and you do the canoe. This is cyclic AMP. Let's keep them separate. Anyway, so there's your cyclic AMP, and that's going to activate a kinase we'll talk about shortly. Now this gets broken down by phosphodiesterase. You can see this is a phosphodiester, double bond O, single bond OC, phosphodiester. PDE is going to come in, break that bond, and now you're back to good old AMP, which will be regenerated into ATP eventually. So there's your cyclic AMP made by so the two enzymes that are important here, adenyl cyclase and phosphodiesterase. Let's make this guy bigger. Every time I go through these slides, I update them a little bit. So I will delete what's up in your Brightspace and change out for this one. Oops. I want to take these figure numbers out too. These figures are from World of the Cell, a different textbook I use. Okay, so how do we make cyclic AMP? We've pretty much got it all put together already, but let's go through it. You've got your receptor. Ligand binds to receptor and activates it. Activated receptor then becomes a GEF, and the GEF will cause release of GDP and pick up GTP. The alpha subunit then releases the beta gamma and binds to adenyl cyclase. Adenyl cyclase makes cyclic AMP. And cyclic AMP then, cyclic AMP then will bind to the protein kinase A. So protein kinase A is composed of two regulatory subunits that are attached to each other and two catalytic subunits. So the regulatory subunits are basically just keeping the catalytic subunits at bay here. When cyclic AMP is around, one molecule binds to each of the regulatory subunits, and that releases the two catalytic subunits, and they go off and phosphorylate proteins and produce the effects we see in the cell. So cyclic AMP then, I guess I didn't show this, cyclic AMP activates protein kinase A. And then the alpha subunit will shut itself off, and again cycle and keep cycling and turning itself on until the ligand's gone. Now we get rid of cyclic AMP with phosphodiesterase breaking it down, and that shuts the mechanism off. So one thing I like to stress is if you really know this system, and you should know which steps in it are catalytic and which ones are stoichiometric. So where are we getting amplification of the signal here? Let me go through it again and talk about that. You should also know how each of these steps is turned off. So, oops, where's the first one? Okay, so the ligand binds to receptor, that's 1-to-1, that's a stoichiometric activation. The receptor then can interact with hundreds of G proteins. Okay, once it's active, different G proteins will come in and get activated. So you've got an amplification step there. Once the G protein is activated, it's going to bind to the effector molecule, and that binding is 1-to-1. So again, that's stoichiometric. But once you activate this effector molecule, it becomes an enzyme, so it is catalytic. So we get amplification of the signal here too. So if we made, if this activated 10 G proteins and this did 100 ATPs, now we've got 100 cyclic AMPs for one ligand. And then the cyclic AMP activating pKa, this is stoichiometric, right? 1-to-1 binding. You release this enzyme, now that's catalytic because it's going to go off and phosphorylate every phosphorylation site it sees. And then how do we turn each of these steps off? Ligand receptor interaction is reversible, right? It'll bind and then let go and come off. As long as you've got ligand out there, another one will bind to it. Once you don't have ligand out there, this stays in the off position. And we already talked about how this shuts itself off by breaking down its GTP. And this gets shut off when we break down our cyclic AMP into AMP. And I'm gonna, we'll talk about that later. Okay, what does this do? I've kind of skewed this figure a little bit to talk about the sympathetic nervous system. Of course, epinephrine is not sympathetic; it is a hormone, not a neurotransmitter. But in terms of the sympathetic, I like thinking about cyclic AMP in terms of the sympathetic nervous system because it makes it easy to remember. What do you want when you're under stress? You want to break down glycogen. So cyclic AMP breaks down glycogen in the muscles of the liver, releasing glucose, more energy out there. Same thing with your adipose tissue. Break down your triglycerides, release fats, more energy to deal with the stress. Cardiovascular system, epinephrine is going to increase your heart rate. It's actually going to decrease your blood pressure. Norepinephrine would increase your blood pressure. But you're increasing blood flow, your cardiac output anyway. And that's what you need in the stress. And this is why your blood pressure goes down. Epinephrine is going to relax the arteries going to your skeletal muscle. And that's going to cause a decrease in blood pressure, but an increase in flow, more blood to your skeletal muscles. And mydriasis in the pupils, right? Your pupils open up wide due to epinephrine or norepinephrine. Okay, so that's it for cyclic AMP. Now, we've been talking about this since chapter 2. Maybe this is the last time we'll have to talk about this. We'll see. But anyway, PIP2 has got the polar head group is inositol with 3 phosphates on it here. Phospholipase C is going to come on, cleave that away. And that's going to release diacylglycerol, which activates protein kinase C, and IP3, which is going to release calcium from the ER. And here's that system in a nutshell. You've got your ligand binding to receptor, receptor activating G protein activating phospholipase C. Now this is an isoform. It's phospholipase C beta. The one we saw with the receptor tyrosine kinases was phospholipase C gamma, just so you know the difference there. And you can also see the little red guy here. That's diacylglycerol. So what is this going to do? I'm glad you asked. We activate the receptor. Oh, and the G protein, I'm sorry, I should have said this. The G protein here is GQ. So it's the subunit, it's the another isoform of G of the alpha subunit GQ. And that's going to release IP3. IP3 receptor, increase calcium in the cell. Diacylglycerol is going to activate protein kinase C. Okay, we've seen protein kinase A. AKT is protein kinase B. This is protein kinase C. And protein kinase C then will activate some cell surface channels and allow calcium to come in. So we get an increase in calcium in the cell, and that's going to activate a bunch of processes. How does it activate those processes? Another protein we've seen before, calmodulin. So calmodulin is a calcium binding protein. If you look into it, this structure is called an EF hand. So you've got an EF hand at either end of this molecule, and when it binds calcium, that causes it to become active. It will then bind to some other protein and turn it on. Now, in chapter 3, we were talking about contraction of smooth muscle. And so there we saw calcium increasing in the cell, activating calmodulin, and then you get myosin light chain kinase being activated by calmodulin. So that's one example. There's also just a calmodulin dependent protein kinase that can get activated. And later we'll see a third enzyme, nitric oxide synthase, that can get activated. So bottom line being, calmodulin's kind of a intermediary protein that's going to activate a bunch of other things. Examples of this, again, here I'm kind of, no, I'm not. This is somewhat parasympathetic, but not really. So thrombin's going to activate platelets, causing them to aggregate and block the injury. Smooth muscle, acetylcholine can cause muscle contraction. Acetylcholine, again, your parasympathetic neurotransmitter, causes insulin secretion, amylase secretion. So rest and digest. You're gonna stimulate your stomach to start digesting, your intestines to digest, and releasing more hormones to help you be a better person, I guess. Okay. All right. Angina. So we're going to talk about nitric oxide synthase next. Let me see. Did I talk about? All right. Let me back up for a second. There's something I forgot to put in the slides here that I really want to talk about. I'm going to bring up a different PowerPoint, and I will update the notes to show you guys what I'm talking about. What in the hell is he talking about? Well, you'll find out, won't you? So this is a different class I teach. Okay, so here's our cyclic AMP system, right? Here's the G protein GS. Another G protein that I forgot to tell you about is GI. So there are other G protein coupled receptors that will activate GI, and GI inhibits adenyl cyclase. So if you've got these two, if you're activating both these receptors, you've got a physiological antagonism here. This is trying to increase cyclic AMP while this is blocking it. So GI is another important G protein. I'll add that to the notes later, and I may put this slide in at the very end, so that you can see that when you want. All right. Anyway, back to nitric oxide. So this is, I love this example because it's an example of nitric oxide being used clinically. So, you know, if you've got angina, it's because you've got plaque buildup in your coronary arteries. So your heart can't get enough blood, and when you exercise, your heart's not getting enough oxygen, so it goes into lactic acid production, and you get pain in your chest. So what do you do about that? You take a pill and you put it under your tongue, and the pain magically goes away. So what's going on is that pill is nitroglycerin. And nitroglycerin is organic, it's nonpolar, dissolves through your tongue, goes into your body, and it's going to produce nitric oxide mainly in the venous side. And so your venous return is going to decrease. And because you've got less blood coming back to the heart, it's working less hard. It can get more oxygen during relaxation, and the angina can go away. And then you're going to get your butt to the hospital and get a little Roto-Rooter job done there. My buddy Perky Go-Go in the cath lab will help you out over there. Okay. So what is nitric oxide? So what I'm going to show you here is the system where nitric oxide was first discovered, and it's has to do with the endothelial cells regulating the arterial smooth muscle cells and controlling the contraction of arteries. And it's kind of an interesting story of how it was found by Fritschgott. Basically, they were doing rabbit ear or rat aorta? Well, they were doing arteries, small arteries, and it was like sometimes when they hung them up, they had added acetylcholine and they'd contract. Sometimes they hang them up and they'd relax, and they're like, what the hell's going on? And what they realized, if they were really, really careful in how they dissected out those vessels, they always got relaxation. If they were rough with it, then they got contraction. So they figured out, oh, it's the endothelial cells. So the receptors that were causing relaxation were on the endothelial cells, and something endothelial, we used to call it endothelial derived relaxing factor, EDRF. We didn't know what the hell it was. So it's being released and causing relaxation. So with time, people figured out it was nitric oxide. So anyway, this is, again, in chapter 8, we were talking about histamine. This is how histamine relaxes vessels when there's a mast cell degranulation. Histamine, here we're saying acetylcholine, which is true as well, but also histamine will bind to receptors on endothelial cells. And this is going to activate that whole IP3 calcium system. So we get breakdown of PIP2, production of IP3, and then release of calcium from the ER. And again, like we just saw, that's going to bind to calmodulin. Now, here's where the pathway diverges. Calmodulin is going to bind to nitric oxide synthase. So nitric oxide synthase is going to take the pirate's favorite amino acid, arginine. It's going to take arginine and convert it into nitric oxide and citrulline. So arginine has got this a lot of nitrogen in its side chain. It's going to take one nitrogen off, react it with an oxygen. There's your nitric oxide. Citrulline is just a byproduct. It just, I think it just gets metabolized or something. So anyway, calcium release, calmodulin, nitric oxide synthase, nitric oxide. And then nitric oxide then diffuses from one cell into another. So it goes into the smooth muscle cell, activates guanyl cyclase. Guanyl cyclase then is going to activate the production of cyclic GMP. Cyclic GMP will activate protein kinase G. And protein kinase G is going to do a lot of the same things as protein kinase A. So they both cause relaxation in smooth muscle cells. So this is how we're getting smooth muscle relaxation with histamine. Goes through that endothelial cell, calcium, calmodulin, nitric oxide, guanyl cyclase, cyclic GMP, protein kinase G. Okay, so that's nitric oxide, and this is what happens in the blood, in the arterial cells. This is a general mechanism you can find in a lot of different cells, and is found in the central nervous system and everything. So nitric oxide is a really important local modulator. So here is physiological antagonism, and this is not in your book, but it's something I like to talk about as a pharmacologist. So in terms of pharmacology, when you think of an antagonist, it's antagonizing at the receptor level. Let me close my door. So, it's going to be competing for the same receptor as the endogenous agonist. Here we're talking about antagonism further down the line. So if you've got like norepinephrine versus epinephrine, right? You think of them both as sympathetic, but they have different effects, really. So your norepinephrine is mainly, oh God. Your norepinephrine is mainly activating alpha receptors. Alpha receptor is going to be linked to GQ, IP3 calcium, myosin light chain kinase, contraction. Epinephrine, on the other hand, is activating a beta receptor. Beta receptor activates cyclic AMP, protein kinase A, which is going to decrease calcium in the cell and cause relaxation. So this is physiological antagonism. Somewhere down the line, they're interacting with each other in opposite ways. Kind of interesting too to look at it. Here's norepinephrine with the free nitrogen here, and epinephrine with a methyl group on that nitrogen. That's why it's called norepinephrine. NOR are 3 German words that mean without radical. So it's epinephrine without that methyl group on it there. Another trivia factoid. Okay, so that is cyclic AMP and IP3 calcium, those systems. Now, another thing G proteins can do is interact with ion channels. So this is an example that we can see in the SA node of the heart, where acetylcholine binds to a muscarinic receptor, which then activates the G protein. And interesting thing about this mechanism is this time it's not the alpha that does anything, it's the beta gamma. So the beta gamma subunit then opens a potassium channel. And they show potassium going either way, which is kind of stupid. Potassium is going to be going out and it's going to hyperpolarize the cell. I'm going to take that out next time I show the slide, because I mean, it can go in, it depends on the membrane potential, but basically it's going out. So this is, I believe we talked about EPSPs and IPSPs. This is one way to do an IPSP in the cell. Let me just get rid of this. I don't like that at all. Oops, hang on. See, potassium is just going out. I don't know why I can't lose that line. Anyway, I'll come back to it. All right, so, okay, so that's it for G proteins interacting with channels. All right, so then the last thing we're going to talk about in this chapter, and this is not in your book. I don't know why it's not. This is really, really important. But this is receptor regulation. So as you probably know, the more you take a drug, the less effect it has, right? And that's because receptors in the membrane are not static, they're dynamic. They're constantly being regulated based on how much stimulation the cell is getting. I heard a pharmacologist once say that the cell doesn't respond to the hormone, it responds to changes in the hormone. So if the hormone stays constant, it stops responding to it. If it goes down, it responds to it. If it goes up, it responds to it. But the cell adapts to whatever stimulation it's getting, and then it's looking for changes. So here's your G protein here in the membrane. When the hormone binds to it, it becomes active, and it then activates some kinases like protein kinase A, but it's also going to activate a kinase called GRK. So GRK is going to be recruited to the membrane by the beta gamma subunit. So here's another role for that beta gamma. Once the alpha subunit goes off and starts doing its signaling, the beta gamma recruits GRK to the membrane. And GRK then is going to phosphorylate that cytoplasmic tail of the G protein coupled receptor. And that's going to cause the recruitment of another protein called arrestin. Arrestin binds to it. Now, interesting thing about that's going to do two things. The phosphorylation and arrestin binding does two things. One is it decreases the affinity of the receptor for the ligand. So mostly that ligand is going to come off. And the other thing it does is it blocks the ability of the receptor to interact with G proteins. So this is desensitization, okay? The cell is becoming less sensitive to the ligand because the receptors are being blocked. Now, if that stimulation stays there longer, this is kind of like a short-term thing. This happens quickly. If it stays there longer, this is going to target that receptor to go down into a calathrine-coated pit. So these kind of triangular-looking things here, or three-sided things, are calathrine. And clathrin is going to coat the membrane in such a way that causes it to invaginate, and it's going to pull that membrane in, and you get endocytosis. So the receptor gets in this pit, it gets endocytosed, and now it's here inside the cell. And arrestin will get taken off, it'll get dephosphorylated, and it will sit here in this subcellular pool of vesicles. So just like that glucose transporter we talked about earlier today, you've got a lot of proteins that are sitting there in this pool of vesicles right under the membrane, so the cell can rapidly respond to things. So if your your ligand goes away, right, while it's there, you're downregulating receptors. When it goes away, that receptor might get put back into the membrane and reestablish the sensitivity of that cell. On the other hand, if you're chronically overstimulating the cell, those receptors are going to go to the lysosome and they're going to get degraded. So over time, you end up with fewer and fewer receptors. And this is part of what makes people go into withdrawal when they're on a drug like heroin, right? So heroin's activating these G protein coupled receptors, and you get rapid desensitization. So pretty quickly, they're increasing their doses and increasing their doses until they're, at really high doses of that heroin or fentanyl or whatever. And the problem with that is you've downregulated all these receptors. Now, if you don't have heroin anymore, the endogenous endorphins you have there aren't enough to activate receptors to keep you normal. So now the cell's going to go into this, oh my God, I don't have heroin anymore. We don't have enough receptors. We're not getting stimulation. Withdrawal, right? So that was what makes you go into withdrawal, the fact that you don't have those receptors anymore, and the endogenous neurotransmitters are not enough to stimulate that cell. And then, over a period of a week or two, those receptors will come back, but you have to go through that withdrawal period before it corrects itself. But I get a kick out of this. My undergrads love this. I don't know how realistic it is, but this is what happens when you take cocaine. So this is over a very short time period here. So when you take cocaine, you feel yahoo. Cocaine is really good stuff. When you don't have enough stimulation through your dopamine system, you feel pretty blah. When you take cocaine, you feel great, but you got to remember cocaine lasts about 30 minutes. So the effect goes away. And when it goes away, you've downregulated some receptors. So you're not feeling back to normal. You're feeling kind of crappy. So what do? Well, you know what they say, rehab is for quitters. So you take more cocaine and you go back up. Now you're not peaking as high anymore because you've got fewer receptors there. You probably have less dopamine to release as well. So you're not getting a peak as high. And when that cocaine goes away, you're down even lower than you started. So now you really want that cocaine. So you take it again, and now you're not getting as high again. You need a higher dose of cocaine to get a better high. And that kind of cycle continues until basically you run out of dopamine and you're just doing that cocaine to keep the norepinephrine stimulation going because you're really not feeling great anymore. And then eventually you're going to stop doing that cocaine and you'll slowly recover those receptors. So anyway, that's kind of the cycle of drug use. So that's it for this bad boy. All right, 12:02. I hope I didn't go too fast. I've gone through everything I wanted to go through. Let me just stop sharing here. It's a problem with these classes. I share too much. That's funny, Dr. Slush. Okay. How we doing out there? Can I see your faces again? Matthew Tyler Coate. There's Tuck. And, oh, wait, who is this? Who is this person? I don't know you. Ah, there's Zoe Albert. David Roy, Patrick Myrock, Hannah. Okay. All right, so that's all I have for you today. One announcement today, My goal for the afternoon is to come up with a test, and I'm going to put the answers in the test this time. So I'll put a practice test up there. Here's your questions, and then here's the questions with the answers. So you know where I'm coming from and what I'm expecting. We'll have a quiz this weekend. I'm through with chapter 9. I don't think we want to keep going until we have an exam. The exam is scheduled for Thursday. How about this? Maybe I should put it to a poll, but how about I open that exam Tuesday and we'll let it go Tuesday through Thursday, because that gives people a couple of days to do it. Does that sound okay? So then we won't have class on Monday because I've run out of things to talk about, and I'm not that interesting a guy. So let's just call it a day on Tuesday. We'll start the exam Tuesday morning, done by the weekend. I'll grade it on Monday, and you'll get your grades back before Tuesday. How's that sound? Good? All right. So that's it for today. Stay hydrated. Talk to you later. Bye.